Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goeie. I am the pro-rector of this ceremony and the chair of the promotion committee. First of all, I would like to welcome Mr. Joël Beaumont. He will defend in public his thesis, which is called Extracellular Vesicles in the Hallmarks of Cancer, Mediators of Intercellular Communication and Beyond. Welcome all members of the degree committee here, in particular the two supervisors, Professor Kasper Roeschop. He is professor of radiotherapy at Maastricht University, and Dr. Tom Keulers. He is associate professor in radiotherapy, also at Maastricht University. And I would also like to welcome all other members of the committee. I will introduce the 6 opponents later during the ceremony, because if I mention all those names now, you will certainly forget. Welcome the audience here in the aula, and I would like to welcome all followers of the live stream. Mr. Beaumont, may I ask you to give a summary of your thesis, and I wish you success in the coming hour. The floor is yours.
Thank you very much. Dear Pro-Rector, dear members of the Corona, dear family, friends and colleagues, welcome and thank you for being here today. In the next 15 minutes, I will give you an overview of the research I performed during my PhD, in which I investigated the role of extracellular vesicles in the hallmarks of cancer. However, before I dive into the specifics of my research, I will give you an overview of what cancer exactly is, how it develops, and what specific characteristics these cancer cells acquire. Now, as you probably know, the human body is composed out of billions of cells, and all of these cells work closely together to form tissues and organs with very specific functions. But how do these cells know what they are supposed to do? They know this because each of these cells contains DNA. And DNA you can consider as the blueprint of a cell. So it tells the cell what to do, where to move, or for example, when to divide. Now, during the development of cancer, this DNA can become damaged, for example, due to exposure to chemicals or smoke. Once this DNA is damaged, the instructions written within it are not understood clearly anymore by the cell, and the cell will turn into a cancer cell. Now, these cancer cells, one of the typical characteristics is that they start to divide uncontrollably. And this generates more and more cells, all with the same damaged DNA. And this eventually leads to the formation of a tumor. And when left untreated, this tumor will continue to grow, thereby destroying the surrounding healthy tissue, eventually leading to death. Now, besides continuously growing, cancer cells also develop other typical characteristics, known as the hallmarks of cancer. First of all, cancer cells consume a lot of sugar, and the sugar is used within the cancer cells to produce the energy which they need to grow. In addition, cancer cells also induce the formation of new blood vessels, and blood vessels provide the tumor with the oxygen and nutrients, again, which it needs to grow. Besides this, also the movement of cancer cells is affected, and where normal, healthy cells mainly stay within a specific tissue where they perform specific functions, cancer cells acquire the ability to move away from this initial tissue into surrounding tissues and distant organs where they can form new tumors. Now, many of these hallmarks of cancer require the communication of cancer cells with their environment. And one of the ways via which cancer cells communicate with their environment is via the production of EV, or extracellular vesicles. EV are small particles released by almost every cell type in the human body. And they contain a lot of proteins and other molecules which are transferred from one cell to another as a form of communication. Now, this is a very normal and healthy process which happens in all of our bodies. However, during cancer, communication via these EV can be disturbed. And it's this what I investigated during my PhD. So how do the EV from cancer cells contribute to cancer progression? Now, communication via EV can also be influenced by various factors, one of which is the exposure of cancer cells to hypoxia, or low oxygen levels. Hypoxia is generated in a tumor due to fast-growing cancer cells and slowly growing blood vessel cells. And due to this rapid growth of these cancer cells, cells located further away from the blood vessels are exposed to lower concentrations of oxygen. Now, hypoxia, unfortunately, also makes cancer cells more aggressive. And for example, hypoxic cells are more resistant to treatment. Therefore, it's very important that we understand what is the difference between these hypoxic cells and cells exposed to normal oxygen levels, so that we can develop better treatments for patients. During my PhD, we have shown that hypoxia stimulates the protein levels of a protein called GABARAPL1 in cancer cells. And we wanted to investigate what this GABARAPL1 is doing in these cells. Therefore, we generated cells in the lab which have either high levels or low levels of this protein. And then we compared the behavior of these cells to understand what this protein is doing. By doing so, we discovered that GABARAPL1 is important in the formation of EV. And as you can see here in this graph, cells with high levels of GABARAPL1 also release more EV. So this also suggests that hypoxic cells increase the levels of GABARAPL1 in an attempt to communicate with their environment. Now, not only the amount of EV produced is important for their ability to communicate, also their content is very important in this function. Therefore, we next also evaluated the content of these EVs produced by these different cells. And here we show that cells with high levels of GABARAPL1 also release EV with high protein content. So from this data, we can conclude, first of all, that GABA-APL1 is very important for the formation as well as the loading of proteins into EV. And besides this data suggests that hypoxic cells increase GABA-APL1 in an attempt to communicate with their environment. Now, why would hypoxic cells try to communicate with their environment? We have shown that these hypoxic cells increase the levels of GABA-APL1, which stimulates the formation of EV, which can induce the formation of new blood vessels. These new blood vessels, in turn, provide the tumor with enough oxygen and nutrients so that it's able to continue to grow. And if we look at a real-life situation, we also indeed see that tumors with high levels of GABA-APL1 also grow faster than tumors with low levels of this protein. And this is due to increased presence of blood vessels. Now we wanted to understand, how is this happening? What are these EV doing? Therefore, we isolated the EV from cancer cells, and we gave these EV to the cells of the blood vessels. And then we measured how these cells reacted to these EV. By doing this, we discovered that these cells increased the consumption of sugar. And as I told you before, sugar is used in cells to produce energy, but it can also be used for various other purposes in the cell. And we demonstrate that in these cells, the sugar is used for the production of amino acids. And amino acids you can consider as small building blocks which the cells need to produce proteins. Therefore, we next also evaluated whether these cells make more proteins. And indeed, we show that these EV stimulate the formation of new proteins in these cells. Now, proteins can be thought of as the workers of a cell. I have told you before that the DNA contains all the instructions for a cell to know what to do and how to function. These instructions are actually carried out by proteins. And in the case of blood vessel cells, these proteins are required for the formation of new blood vessels. And indeed, we also demonstrate that these EV stimulate the formation of new blood vessels. So to summarize the first part of my thesis, I have shown that cancer cells release EV, which are stimulating the consumption of sugar in blood vessel cells. This sugar is used for the production of amino acids and proteins, which are required for the formation of new blood vessels. And these blood vessels, in turn, provide oxygen and nutrients to the tumor so that it can continue to grow. Now, in the second part of my thesis, I investigated the development of metastasis, also known as the spreading of cancer cells throughout the body. As I told you before, movement of cancer cells is affected, and cancer cells acquire the ability to move away from their initial site into surrounding tissues as well as distant organs. And patients which are initially diagnosed with a breast tumor can also develop tumors in their lungs. Now, the development of metastasis and the spreading of cancer cells throughout the body is one of the leading causes of death in cancer. And the development of metastasis is not a really straightforward process. First of all, cancer cells and tumors are surrounded by a layer of proteins, and cancer cells need to invade through this layer of proteins in order to be able to invade into surrounding tissues. Then, once this cell is invaded into surrounding tissues and it encounters a blood vessel, it needs to invade into this blood vessel, migrate through the bloodstream to distant organs, and then again invade through this blood vessel wall to be able to form a tumor in a distant organ. Previous research has shown that hypoxia stimulates the development of metastasis. And the higher the level of hypoxia in a tumor, the higher the chance that your cancer will spread throughout your body. Now, since we know that GABA-APL1 is important, is increased in these hypoxic cancer cells, we wanted to see whether it also plays a role in the development of metastasis. So to investigate this, we injected tumor cells with either high or low levels of GABA-APL1 into the breast tissue of mice. And then we evaluated how many tumors were formed in the lungs of these mice. By doing this, we found that tumors with high levels of GABA-APL1 also form more tumors in the lungs of mice. so indicating that indeed GABA-APL1 is important in the development of metastasis. Now again, we wanted to figure out how is GABA-APL1 doing this. And since we know from the first part of my thesis that GABA-APL1 plays an important role in the formation of EV, we decided to take a closer look into the composition of these EV. And by doing so, we discovered that EV are filled with enzymes. And enzymes you can think of as little scissors. And when these scissors come in contact with a big protein, they are able to cut down this protein into small, tiny pieces. Now remember that earlier I told you that tumors are surrounded by a layer of proteins. We demonstrated that if you add EV to this protein layer, that these EV are able to break down the proteins in this layer. This, in turn, stimulates the invasion of cancer cells through this layer and into surrounding tissues. Now, since we have demonstrated that GABA-APL1 plays an important role in the development of metastasis, we also wanted to determine whether GABA-APL1 is involved in the production of EV, which can stimulate the invasion of cancer cells into surrounding tissues. To investigate this, we again cultured our cells with high or low levels of GABA-APL1 and isolated the EVs of these cells. And then we used these EVs to treat this protein layer surrounding the tumor. And indeed, we demonstrate that EV coming from cells with high levels of GABA-APL1 also stimulates the invasion of cancer cells into surrounding tissues more than EVs coming from cells with low expression of GABA-APL1. Collectively, we have shown that hypoxic cancer cells increase the levels of GABA-APL1, which produces EV able to break down the protein layer surrounding a tumor and thereby increases cancer cell invasion into surrounding tissues and the development of metastasis. So to summarize my thesis, I have shown that EV play an important role in the hallmarks of cancer. First of all, these EV stimulate sugar consumption in cancer cells, in blood vessel cells, and the sugar is used for the production of amino acids and proteins which are required for the formation of new blood vessels. And these blood vessels, in turn, provide the tumor with oxygen and nutrients to grow. In addition, cancer cells release EV, which are able to break down proteins surrounding the tumor, thereby stimulating the invasion of cancer cells into surrounding tissues and the development of metastasis. And with this, I would like to conclude my presentation. Thank you very much for your attention, and I would like to give the word back to the pro-rector.
Thank you, Mr. Beaumont, for a clear summary of your thesis.
You're welcome.
The opposition will be opened by the chair of the assessment committee, Professor Theodor Kok. He is professor of toxicogenomics at Maastricht University. Professor Kok.
Thank you. Dear candidates, congratulations on the finalization of your thesis, in which you describe an impressive range of studies in order to molecularly better understand the mechanisms of how these extracellular vesicles from hypoxic cells may contribute to tumor progression. And I also include your promotion team on this impressive amount of work. And the first time I read your manuscript as a member of the committee, as the chair, which is, by the way, something like a half a year ago, if it isn't more, I wrote that this thesis is very well written, easy to read, and after half a year, I have to come back to that last bit. It's still well written, it's easy to navigate, but hell no, it's not easy to read. And I have to do a lot of thinking and processing of the data and all the data that you present, and in order to make sense out of it. So the compliments were to you and your committee. The questions are for you. So let's go to chapter 3. On page 73, you introduce H220L cell type and the ECRF24 as cells of your method. And these drop out of the sky. You don't introduce which one is the EC producing cell type or which one is actually the receiving cell type. You just describe how you culture them. Mm-hmm. And so I was looking for confirmation, because that's what you do, and start reading. And I started wondering, why do you choose these cell types in the first place? I went looking for that information, also couldn't find that. So please, can you elaborate a little bit on the choices of these specific cell types?
Of course. Highly esteemed opponent, thank you very much, first of all, for your kind words and for your question. So these cells, well, basically this whole chapter, came to existence because actually of a question of a reviewer for the paper which we submitted for chapter 5. And the question of the reviewer was, okay, it's very nice that you show that EVs are taken up, but do the contents of these EV actually also end up in the cells and in the different compartments? So starting from there, we actually developed this new method to trace the cargo of EV in recipient cells. So that's the reason why we chose these cells, because that were the same cells which we were using in chapter 5.
Yeah, but does that then also imply that you, and therefore I also have to assume that this is a representative cell type to draw these conclusions, that would apply to any other cell type?
Well, in the end, we will have to repeat this experiment for any setup that you're investigating. So for me, I was interested to see how EVs from cancer cells affect recipient endothelial cells.
Okay, but then you're a man of little words, I came to the conclusion, and you have very general conclusions. Like, let's take the title of chapter 4. The title of chapter 4 is the secretion of extracellular vesicles during hypoxia is dependent on GABA-RAPL1.
Yes.
Point. No, it doesn't say in which cell type. You make that a general conclusion. Are there any is there any other evidence from literature maybe or other studies to substantiate that you can draw that broad type of conclusion?
So in chapter 4, we actually used 3 models to demonstrate that GABA-RAPL1 is important. Of course, more research is needed to validate whether this is the case for all cancer cells. But to be honest, I also don't think that this might be applicable for all cancer cells, because the role of GABA-RAPL1 seems to be, if you look into literature, a bit contradictory, because there's people describing pro-tumorigenic and anti-tumorigenic effects. So I do think that at least for these models, indeed GABA-RAPL1 is very important, but I do think more research is needed to investigate why there's this dual role of GABA-RAPL1 in different types of cancer, it seems.
Okay, so a little bit more prudent formulation of a conclusion may be in place. So we agree on that. So if we go back to chapter 3 again, on page 78, you describe that the tracking of the extracellular vesicles derives proteins in recipient cells. And at the end of the very first paragraph, you refer to figure 2C. You write that, as expected, subcellular distribution of labeled proteins was altered after cells were given 24 hours to process the EV-derived proteins and confirm escape from endolysosomal degradation. So if I look at figure 2C, I'm wondering what am I expected to see. So that was on page 78. So, and if I then look at page 78, I see in that figure, I have to make the comparison between the left and the right. I see 2 pretty much the same comparable protein expression profiles. I really have to look into details to find any differences, and you don't describe that at all. That makes it difficult to read. So my question here is, isn't there a better way to make that more quantitative? Could you have, for instance, made that a more quantitative analysis in which you can have a figure to say, okay, this is this percentage up or down, and to make a less individual subjective interpretation possible?
So indeed, overall, the big picture in this figure, let's say, indeed resembles much more, they resemble each other. But if you look closely to, for example, lane 2, you see that the proteins in that band are significantly down. Well, significantly. There's a reduction in the amount of proteins, and there's other bands being formed. So indeed, we could also quantify the individual lanes and determine protein levels based on that. We could also, what we are actually also still planning to do, is do purification of the labeled proteins and then send that for mass spec, of not necessarily purification, but isolate these labeled proteins, label them with biotin, and send them for mass spec analysis, and then you can actually do a quantitative and qualitative comparison.
Oh, it's clear that you have given it some thought on how to make that more quantitative, so that's good to hear. And how does that then confirm the escape from endosomal degradation? Because I would argue if it escapes this degradation that you would not see any alterations. So where does the confirmation come from?
Well, I would hypothesize that if these labeled proteins which are delivered via EV don't escape the endosomal pathway, that they eventually end up in the lysosomes and are broken down. So then you would not be able to detect any labeling. And the fact that we see, for example, signal in the how's it called? The nuclear fraction indicates that at least some of these proteins escape this pathway and are being used, in this case, in the nucleus or in other cell organelles.
Okay, thank you. I have a million other questions. I'm looking at the prorector and he says I'm done. So thank you for responses. I will give the board back to the prorector.
Thank you, Professor de Kok. The next opponent is Professor Guido Jenster. He is Professor of Urology at the Erasmus Medical Center in Rotterdam. Professor Jenster.
Thank you, Prorector. Dear candidate, I also would like to congratulate you and your whole team on coming to this thesis with a lot of fun work. I do like challenging complex figures. I do like puzzles, so this was a wonderful thesis for me to read and to understand. I love it. First simple question. Is GABA-RAPL1 an oncogene?
Highly esteemed opponent, thank you very much for your kind words and your question. As far as I know, it's not described as an oncogene. Because also, again, there are people describing actually anti-tumorigenic roles of GABA-RAPL1, depending on what cancer type they're looking in.
That's a short answer. It does have an effect, clearly, if you knock it down on metastases and many other processes that are absolutely cancer-specific. So, if it is not an oncogene, then how would you describe the function of this gene and all the effects that you have seen?
So the function of this gene, at least for what we show, is in the endosomal pathway involved in maturation of endosomes. It's involved in secretion of EV. It's also involved in trafficking of receptors to the membrane, for example. So there's a lot of functions attributed to this protein.
Right. Yeah, I'm going to interrupt now. That's amazing. Okay. But so for cancer cells, this is all about, right? So it is an absolute advantage for a cancer cell to have high expression of this protein, isn't it?
In our setup, yes.
Yeah. So what can cancer cells do in order to get that protein up? And do we see that in patient samples?
So first of all, one factor which we show that really triggers the expression of this protein is hypoxia.
Sure, that's regulation, but the gene itself?
The gene itself has also been described to be inducible by estrogens, for example. Correct. Initially it was discovered as an estrogen-inducible
but
protein.
That is environmentally controlled. Cancer cells don't really change their environment. So what can cancer cells themselves do to get this protein upregulated?
Probably there are transcription factors which could be hyperactivated or repressors which can be downregulated or mutated in cancer cells. But I have...
What about DNA level? You can just amplify the whole gene, right? Or the gene region.
Also, yes.
Is that happening in cancer samples? Do we see an amplification or even mutations that upregulate the activity of this protein?
To be honest, I didn't look into that.
I can tell you the answer. Barely.
Probably yes.
So it's 5%. It's actually not a bad number. 5% of all cancers, many cancers, have amplified this particular gene. There's also quite a few cancers that have deleted the whole thing, so that's making life complex. But it's not that much, and mutations are very rare. So if you hear this, what does that mean to you if you interpret the role of this protein in cancer and how cancer cells can affect this particular gene?
I would say that this protein plays an important role in the development of cancer, or in some cancers actually in the suppression of cancer. However, it doesn't seem to be essential for the development.
Yeah, I would argue again, like you said in the beginning, it's probably not an oncogene, but it could be a landscaper type of genes or other types of genes definitely affecting cancer cells and cancer processes. So you already mentioned the other name for this protein is, I've got to say this right, early estrogen regulated protein. And the protein itself seems to translocate the androgen receptor to the nucleus, right? So it has an effect. First, it's affected by the estrogen receptor pathway, and it might affect androgen receptors. So I'm just wondering, in all the experiments you have done, do you see differences between female and male cell lines, animal models? Is there anything known about male-female specific effects of this protein on carcinogenesis?
We didn't evaluate this, but I would hypothesize, because for example, in the in vivo model which we used the MDA MB231 cell line, which is negative for the estrogen receptor. I would hypothesize that if you use other cell lines which have the estrogen receptor and you would implant that into female mice, for example, that this would aggravate the effects of GABA-APL1, since estrogen can induce the expression, and we see that that higher expression levels correlate also to worse outcome. But we didn't specifically check for this, no.
Yeah, so indeed, in studying that part, you took the 231 cell line, you go for estrogen receptor positive cell lines, you might actually see a potential difference. What about on the male side, if the GABA-APL1 actually gets the androgen receptor into the nucleus and maybe activate the pathway stronger in that respect? How would you study that?
Sorry, how
would you study
exactly?
the male side, so the testosterone and androgen receptor pathway
like
and the effect of your protein on that particular pathway? Which model system would you use and which manipulations would you give it a try to find out whether this protein might actually have a role there?
Well, you could use male mice, investigate if indeed like nuclear translocation of these receptors is indeed increased in these cells, or decreased if you knock down GABA-APL.
Which cancer type would you study?
Probably prostate cancer or something
Wonderful.
in that direction.
That is the right answer. Guess what I'm doing? Well, anyway, I would like to thank you very much for your fun answers and discussion. I would like to give the word back to the prorector.
Yeah.
Thank you, Professor Jenster. The opposition will be continued by Professor Rory Koenen. He is Professor of Biochemistry at Maastricht University, and he is also Secretary of this committee. Professor Koenen.
Thank you, Mr. Prorector. Yes, dear candidate, also my compliments to you and your PhD supervisory team for this very nice and very elaborate thesis. And I was sort of intrigued by the whole concept of hypoxia, because well, it's not supposed to happen in normal physiology. And I've been looking on PubMed a little bit of what would be the physiologic function of hypoxia, and it all comes down to tumors, so you can't even separate it. They're all talking about tumors. But hypoxia triggers a complete program of genes with hypoxia-inducible factor 1 alpha and all this. So hypoxia and this whole mechanism cannot just be there to promote tumor growth. So what do you think is the physiologic purpose of this whole hypoxic program that is hidden in the normal cells?
Hi, esteemed opponent. Thank you very much for your question. So first of all, indeed, hypoxia, it does occur in normal physiology. For example, in the bone marrow or cartilage, they are hypoxic. However, besides that, hypoxia is a relatively specific factor of tumors, indeed. It doesn't, its role, well, how do you say this? Its role is not necessarily to stimulate cancer progression, but it's there because of the high proliferation of your cancer cells and of the metabolic needs of these cancer cells, and this generates, in the end, hypoxia. In turn, because of the upregulation of HIFs, which in normal physiology restores oxygen flow to the tumor to the normal tissue to restore normal physiology. In tumor cells, this actually indeed provides the tumor again with oxygen and nutrients to grow. Plus, during hypoxia, also protective mechanisms are activated, which prevent the cells from dying during hypoxia. However, in cancer cells, that means also that these cancer cells are more resistant to for example, treatment due to upregulation of autophagy.
Okay, thank you. I just so also in your answer, I see you cannot get around cancer to explain the role of hypoxia, which is, I think, simply very, very difficult to do. And but I thank you because there is indeed hypoxia in the normal body, and I think angiogenesis is probably one of the systems that is triggered by hypoxia that needs this inherent program. Okay, so. This was sort of as a bridge to chapter 5, because in chapter 5, and you also presented that in your introduction, you treat endothelial cells as sort of the vessel cells with EVs that are derived from different tumors. And these tumors are different cell lines, tumor cell lines, and in turn they are made hypoxic to different extents. And then you see what happens. So on page 144 to 146, that's figure 3, you see you measure effects on lactate production, glucose uptake, and very elaborately. And I have to say, and maybe you agree, that the effects you see really don't knock someone off their feet. At least they didn't knock me off my feet. The cells really don't appear to care whether they're hypoxic or normoxic. So I just looked up a review, and then there was this beautiful word of Warburg effect. And yeah, for me, just explain the Warburg effect to the audience. That is that tumors, they have a sort of a hypoxic type of metabolism every time. They don't really care in their metabolism. They just use this glycolytic metabolism, whether there's oxygen or not. So I was just wondering, what is the effect of the Warburg effect in the cells that you use? Maybe they don't care whether there's oxygen or not, because they are sort of primed to an, hypoxic environment anyway, whether there's oxygen or not. So might that be the reason why you see so little effects between the normoxic and the hypoxic cells? And what is the effect of Warburg effect in general in your whole research?
So indeed, the Warburg effect is that glucose is primarily broken down in glycolysis to produce ATP in cancer cells. However, that's only one aspect of hypoxia, which would be induced by hypoxia. Hypoxia induces glycolysis, but there's also many other factors still upregulated, deregulated, once the cells are exposed to hypoxia. Now, the Warburg effect is just the fact that the metabolism part, at least, resembles the hypoxic metabolism. That being said, indeed, there's not a lot of difference between normoxic, hypoxic, and anoxic EV. However, if you look further downstream in the pathways, you do see that somehow the usage of glucose downstream is determined by the hypoxia levels. So,
okay. Great. Let's keep it there, because that is a perfect transition to a question that I have about your figure 4. And I'm going to skip other questions for the sake of time. I hope I'll be in time. So in figure 4, you use a very nice experiment, mass spectrometry with isotope labeled glucose, and then you see which amino acids are being produced. And especially figure 4H jumps into my mind. That's the endothelial cells that were treated with the U87 cells. And in figure 4H, you see 2 amino acids that are red, and luckily I can see red and green. So L-glutamine, or glutamic acid, sorry, and aspartic acid.
Yeah.
Do these amino acids ring a bell to you? Or can you imagine why they spring into my mind?
I've heard of them, yes.
Yes, okay.
But I don't know where this question is leading.
These are the amino acids that are sort of related to anaplerosis. And anaplerosis is the feeding of the citric acid cycle with pyruvate and with ketoglutarate, and I think it's oxaloacetate. They're coupled in. So if they accumulate in the cell, couldn't you say that the TCA, the citric acid cycle, is simply halted under these conditions?
I get the reasoning, but this is H, eh? Oh no, that's intercellular levels. Okay, that could very well be, yes. However, and I have to recall the data of 3 years ago now.
Oh, good.
I don't think we saw differences in the TCA cycle in terms of activity.
Because that was my question that I wanted to ask according to that. Okay, I'm very happy with your answers. I'm going to give the word back to the prorector.
Thank you, Professor Koene. The next opponent is not here, but she is online present here in the committee. Dr. Nolte Het Hoen, and she is Associate Professor of Extracellular Vesicles at Utrecht University. Dr. Nolte Het Hoen, the floor is yours.
Thank you very much, Mr. Prorector. And first, I would like to compliment the candidate on the completion of this thesis, and of course also the supervising team. It's a very extensive work, and I think it contributes a lot of important knowledge, both for EV biology, but also on the function of tumor-derived extracellular vesicles. And I would like to start our discussion with a bit of a high-over, more general question. Because when I look at the different chapters in your thesis, then when you study GABA-APL functions in different cell types under different conditions, very often you don't see only one effect. It seems to be that the effect differs a little bit per cell type, per condition. And I was just wondering, what is your vision on what that tells about this protein, this important protein, GABA-APL1?
Very much for your question. So indeed, the effects of GABA-APL1 seem to fluctuate a lot. My hypothesis behind this is that there's different conformations of GABA-APL1. So it has been described in literatures that there's different conformations, but for the rest, there's very little knowledge about these conformations. So my hypothesis is that something somehow is triggering different conformations depending on the tumor type or depending on oxygenation of the tumor, or maybe other factors still, which is probably also functionally altering GABA-APL1, and thereby we see different effects of this protein in different environments. That would also, for example, explain why indeed we see in, I think it was breast, I'm not sure which tumor it was anymore, but there's publications showing indeed a pro-tumorigenic effect of GABA-APL1, which is us, and there's people also showing the opposite. So that could also...
Okay, but if you say about these conformational differences, does that mean that maybe the localization of the protein is different in those cell types? And have you seen something like that? I know you've also done microscopy quite a lot.
So that could mean different things. It could be the localization. It could also be GABA-APL1 is also conjugated to PE, for example, when it's targeted to vesicles. So it can also be that, for example, the PE conjugation does not work anymore. It seems to be involved in trafficking of, well, it is involved in trafficking of receptors to the membrane. So maybe also just a conformational switches the direction of movement, for example. There's many different aspects to this, but which one is in fact working, I don't know. That's
okay. I see work for the future researchers then, I guess. Yeah. That said, I would like to go then to chapter 7, and here you work on the potential role of extracellular vesicles in the modeling of extracellular matrix. And we know that might be important during cancer progression. You also nicely explained that in your introductory talk. And the idea that you propose is actually that matrix degrading enzymes are present on the surface of vesicles, and that may be the reason why vesicles only could degrade matrix. Well, I know there's a little bit of other literature about this, and I think it's quite contradictory what people say about it. And well, you are quite firm in your conclusion that you think that the EVs really degrade the matrix. And I would like to challenge that thought a little bit. And that I would like to do based on the figure 1 in your chapter 7. Okay, here you use a layer of matrigel, and then you add EVs, and then you do a Comassi staining to see whether that changes after the addition of EVs. You see a change and you say, oh yeah, really, this is must be degradation. And I would like to challenge that conclusion by saying, it could also be binding of the EVs to the matrix. And because EVs are very lipid-rich, this may hamper some of the Comassi staining, and that gives the reduction that you see in your graph. So how would you be able to exclude that it's not binding, but instead it is degradation?
That's a very good point. And there's also still other aspects which were in the back of my head, which might explain reduced staining intensity here. We could, for example, also repeat this experiment and then stain for CD markers, EV markers, or put it for mass spectrometry, not mass spec, TEM analysis to see if there's indeed binding of these vesicles to this matrix. However, in terms of concentration, what we used a coating of 2 milligrams per ml of ECM, I believe. And the EVs we use to stimulate this is 1 microgram per ml. So that's a complete different order of magnitude of concentration. So that, to me, would suggest that, well, no. Sorry, this was an invalid answer.
I'm curious what you wanted to say here.
So we could do stainings, we could do analysis via TEM, and, for example, to image if there's vesicles bound. Or we could even stain with lipophilic dyes then, indeed, because you could stain the lipids of these EV. So in those ways you could...
Can I interrupt you? So have you ever tried non-tumorigenic EVs, which may not probably have this kind of degradatory enzymes on their surface, and see whether you saw no effect on the Comassi?
Your question was whether we used non-cancerous EVs?
Yes.
No, not yet. However, it's on the to-do list, because also in chapter 5, we used non-cancerous EV to verify indeed that the effects which we were seeing are due to cancer and not due to just a general EV effect. So it was definitely still in the planning, yes. But we didn't do it.
Yeah. And how do you think, if it was adhesion, would that also explain your effects on the cell invasion, the effect on cell evasion that you see? Is there a possibility that that could also be explained by adhesion?
So indeed, there is also literature showing that besides altering ECM, EV can stimulate migration of cancer cells just by binding to the environment and basically serving as cues for the cells to adhere to. So that could also explain indeed the increased invasion that we're seeing. So indeed, also for that, I would like to verify that indeed there's no binding of your EVs to the matrix. And if that's the case, I would like to see if this indeed is the reason why cancer cells would migrate or invade more.
Right, yeah. Maybe a last small question. In the rest of the chapter, you also focus on LOX proteins. And I was wondering what motivated you to study those and not, for example, metalloproteases or catepsins. Why specifically these proteins?
Because we had data that indeed LOX proteins, first of all, were present in the EV. And because LOX is very well described to play a role in cross-linking of ECM and promoting thereby cancer cell invasion and migration. So that's why we looked more into those proteins. But if you look indeed at figure 4, we see that in LOXL2 at least is not involved in these effects. So in the future, I would like to compare the composition of these EV and GABA-APRIL knockdown EV and see indeed which enzymes are even more in there, or no, not more in there, which are less loaded into these vesicles of GABA-APRIL knockdown cells, to identify indeed which enzymes are responsible for the effects. We do have mass spec data on these HE29, UAD7, and MDA-derived EV, And we did also do a cross-reference between these data sets to identify which enzymes are present in all 3 of these EV isolates.
We have to move to the next opponent.
So we have made knockdowns.
That's great to hear. So thank you very much for the discussion, and I give the word back to the co-rector.
Thank you very much, Dr. Nolte het Hoen. The next opponent is Dr. Veerle Melotte. She is Associate Professor in Pathology at Maastricht University. Dr. Melotte.
Thank you. Dear candidate, of course, I also want to congratulate you on your thesis and also your supervision team. And I also think, or I'm convinced that based on that you were able to finalize this nice thesis, that your supervision team cannot call you a prutser anymore, as you acknowledged in the acknowledgement.
You'd be surprised.
But yeah, to my question. So you discussed already a lot today that you have different outcomes regarding EVs on the cancer, depending cancer context, cancer type, also depending the hallmarks. I was very happy. So you used several cell lines in your thesis, throughout your thesis, and I was very happy as a colorectal cancer scientist that you also used HT29. And I was wondering, I miss a little bit, and I could have done it myself, but I was a little bit lazy. Like, what are your major conclusions regarding EVs in the context of colorectal cancer? And then specifically, also maybe, is there room for EV-targeted therapy in colorectal cancer?
Esteemed opponent, thank you very much for your question. So in terms of targeted therapies, there's definitely room based on our results. So we actually show also in chapter 4 that these GABA-APRIL-dependent vesicles also produce vesicles which have GABA-APRIL1. No, that these GABA-APRIL1 dependent vesicles also contain GABA-APRIL1 on their outer membrane, and that you can actually target these vesicles with EV, and this inhibits, for example, their angiogenic effects. So that's definitely a very interesting path to look further into.
Yeah, maybe to interrupt already there. So you also, in the review, discuss also the role of EVs on immune cells, immune modulation. Are you aware, or is there literature, you don't describe it, at least as far as I know, in your thesis, but is there literature about, for example, the role of EVs in immune cells in colorectal cancer? And is there an opportunity for turning cold tumors into hot tumors, maybe, for immune therapy?
There's a lot of literature on the EV function on immune cells. So a lot of papers showing that immune cells are actually suppressed by the production of EV by the tumor. In terms of therapy in this case, well, I'm going to go far here. You could, if we eventually figure out what GABA-APRIL1 is doing and if it's indeed with the different conformations, you could potentially also target this GABA-APRIL1 if we find out why it's having different functions. And for example, induce a conformational switch if we find out what's driving this switch, and thereby actually change the function of hopefully these EVs from a pro-tumorigenic into an anti-tumorigenic one. So that's an option.
Okay. Maybe I can go then also to my second question. For that, I want to discuss with you proposition 2, and maybe one of the paradigms can read proposition 2.
The difficulty in developing cancer treatments lies not in discovering how to kill tumor cells, it lies in doing so while saving the billions of healthy cells performing vital functions.
Yeah, so therefore I interrupted you because you talk about targeting these EVs in cancer. I missed a little bit when reading your thesis knowledge about EVs in normal situations, so for healthy organ function, especially in the GI. There are a lot of vesicles we know for GI functioning. But how can you distinguish normal EVs from cancer-associated EVs? Is that cargo differences or?
So we have preliminary data actually, looking at the GABA-APRIL1 positive vesicles in the blood of patients, that the amount of GABA-APRIL1 positive vesicles is increased. So it is increased. It's not that in control individuals there's no GABA-APRIL1 positive vesicles. However, we don't know what the function of these vesicles is in normal physiology.
But is that known in general context? Like, okay, you have EVs and you have tumor-associated EVs. The cargo is always different, or there's always more in the cancer situations, or what is the major differences in general, like maybe outside GABA?
Between normal tissue-derived EVs and cancer EVs?
Yes.
So yeah, the cargo for sure is different, also because cancer cells are messed up in general compared to normal cells, so there's a lot of other proteins present in the cancer cells, but because of that also in EV. So there's differences in cargo, there's differences probably in functionality. However, proving that these normal tissue-derived EVs have a function is rather hard, because working with normal tissue in general is not that easy, because they don't grow so fast, they don't produce as much EV as cancer cells.
So still lots to do on
so
Conferring.
yeah, definitely.
Yeah, because if I have a time, okay, because like what I really kept questioning when reading your thesis is, why do cancer cells use this complex way of developing or making an EV, while the cargo can also, for example, in the context of tumor in the matrix, for example, all these proteins which are in these EVs can also be secreted in the classical way. So I was really wondering, like, why would a cell do this? I do understand for long distance communication, but you also really discuss at short distance. So why? What is the yeah?
I think it just also
You may
serves.
give a short answer, please.
Okay, I'll try. So it has been shown that indeed encapsulating the cargo in EVs also protects them from degradation, so it would also be a more permanent and more stable way of communication. But, well, there's, how do I say it?
Lots to do.
Lots, yeah.
Yeah, future students. Okay, thank you. I'm happy for your answers. Thank you.
Thank you, Dr. Melotte. The sixth and last opponent is Dr. Frank Stasse. He is Associate Professor of Microbiology at Maastricht University. Dr. Stasse.
Dear candidate.
You please switch on your microphone.
Turn on my microphone. Excuse me. Dear candidate, also from my side, of course, congratulations. Excellent thesis, well written on a current and relevant topic. And also, of course, congratulations to your supervising team. But of course, we're not only here to praise you, but also to further elaborate and clarify a few ambiguities that are in your thesis. Many of the points have already been discussed during this almost 45 minutes by my colleagues, but still there are a few points that I want to discuss with you. I want to take you to chapter 3. We already discussed some part of it. In this chapter, you describe a new method to trace, visualize, and identify IVs, even when they have been taken up by the recipient cell. In this method, you replace methionine, an amino acid, by an analog, the LHAHA, in cell culture. This analog is then taken up by the cells, and it replaces the methionine. Once in the recipient cell, you can demonstrate the presence of this analog, and therefore the proteins originate from the EVs. You can identify them by this click methodology. You can also show that they are present in several subfractions. At least that's what you claim. The question now is, how can you be sure that this analog that you demonstrate is actually still part of the proteins originally present in the EVs? Because the EVs, after all, after absorption, often they end up in the lysosomes, and it's not inconceivable that the proteins are broken down there, and that the amino acids are released into the cytosol, being recycled there by the protein metabolism of the cell. So you don't know whether they are still part of the proteins introduced to the cell coming from the EVs, or whether they are being recycled by the recipient cell itself.
Esteemed opponent, thank you very much for your question. So indeed, it's possible that labeled proteins from EVs are being recycled within the recipient cell, and thereby free soluble LAHA is set free and incorporated into proteins. However, these experiments have been done, so the uptake experiments have been done in the presence of methionine. I believe it was 100 micromolar, but that doesn't matter. But methionine outcompetes LAHA for incorporation of incorporation in newly synthesized proteins. It has been shown actually that even having 10 times higher concentrations of LAH than methionine, methionine is still preferentially built into these newly synthesized proteins. So therefore, we can, with quite great certainty, say that what we're measuring is indeed EV-derived proteins rather than recycling of LAH within the recipient cells.
Okay, so you're quite convinced, and you will prove this in the future. Because my other question is, how can you identify then these proteins? But you already answered that by mass spec. My other question is about the first proposition. Maybe somebody can read this.
As we learn more about cancer, it becomes more evident that the road towards a cure is longer than anticipated, potentially even endless.
So, these final 3 words of this proposition are not really optimistic.
No.
However, and I looked it up, if you look at the data, it does seem that we have made some progress. In the US, for instance, cancer-related deaths have dropped in the last approximately 20 years from almost 200 for every 100,000 residents in 2000 down to 140 approximately in 2022. So it really seems that despite cancer being one of the leading causes of death, we still don't know how to treat various types of cancer, we're still making significant progress, at least in the treatment of cancer. So why this pessimistic view in cancer treatment, and how do you think we can break this negative view of you and maybe turn it into light at the end of the tunnel?
So we could argue if it's pessimistic or realistic. But what I mainly want to say with this proposition is you never know how far you are away from a cure. So 25 years ago, the first paper of the hallmarks of cancer was published, and we had 6 hallmarks, and we said, okay, this is cancer in a nutshell. When we target all these 6 aspects of cancer, we have it solved. But the more you investigate and the more you learn about a disease, of course, also there's more improvements and we find new things to treat it. But also the more we discover that there's pathways we didn't know of before, which complicate eventually the development of therapies, for example. So basically, until we're at the end of the road, we don't know where the end of the road is. How we would break this cycle, more research, applying for grants, and keep on going until we find like the golden bullet, basically.
Well, don't ask NIH for grants, eh? Maybe one final question. I mean, so we have to do more research. Your thesis was mainly on GABA-APL 1. Mm-hmm. Do you foresee a role in modulating the expression of this GABA? I mean, you showed some very interesting data. I mean, the fact that if you inoculate mice with a GABA-APL knockout and you see this 84% reduction in metastasis, this was really astonishing for me. So do you foresee a role for inhibiting or knocking out or whatever GABA-APL 1 in the future in terms of treatment of cancer? Because I mean, it's a very pleiotropic protein. It has so many different functions. So if you knock out one aspect, maybe you also interfere with the other one, which has a deleterious effect. So I didn't see a direct therapeutic avenue for this.
Well, I definitely think that it's worthy to look into this more.
Mr. Beaumont, as you see and hear, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis, and in particular the quality of your defense. And please await our return here.
Thank you.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Mr. Beaumont, the committee here present has assessed the quality of your thesis and the quality of your defense. And in view of a positive verdict and taking into account the previous diplomas that you have acquired, the committee has decided to grant you the degree of doctor. And Professor Roeschop is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. I invite now your supervisor to take the floor.
All right.
Professor Roeschop.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Joël E.J. Beaumont, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Correctly written.
Joël, Dr. Beaumont. First of all, congratulations with this incredible milestone. And none of us expected anything less than what you have accomplished today. And I think we can all agree you have done an outstanding job, not just today, but also the past 4.5 years. I remember when you first joined us, we had a position available in our group to study EVs in cancer. And after reviewing numerous candidates, you stood out immediately. You earned your degree in biomedical sciences at Maastricht University, where you graduated magna cum laude. And after your master's studies, you already got interested in EV research because you started working as a research assistant to study the role of EVs in multiple sclerosis at the same university. But on top of your strong academic background, you struck us as a kind and pleasant person. And although you are quite a bit shy during these first meetings, we knew right away that we didn't need to look any further. And time has proven that instinct was absolutely right. Over the past 4.5 years, we watched you grow into the strong, creative, independent, and technically very skilled scientist that you are today. And with 4 out of 6 thesis chapters already published in very nice journals, you have every reason to be immensely proud. During your PhD, you made essential contributions to ongoing projects and initiated several from the ground up. You provided valuable insights into how EVs stimulate glycolysis in endothelial cells and other cells, but also pioneering a new method to trace EVs in recipient cells. But beyond viewing EVs as vehicles for intercellular communication, you really thought outside of the box and saw them as entities with their own enzymatic properties, able to remodel the extracellular matrix. A very interesting project you led from the start with very interesting results so far. Your work received recognition nationally and internationally. Your work was selected as a late-breaking abstract, which allowed you to present the data at the ISAF meeting in Lyon, in France. And based on your research, you were invited to write a paper on extracellular vesicles for the prestigious Wolfsburg meeting. And despite a few stressful weeks, you took the lead, coordinated tasks within the group, and ensured its completion with success. At the Wolfsburg meeting later in Oslo, you presented the findings, which were published later that year. But what is most striking to me is how you accomplished all of this. I don't think I've seen anyone ever who earned a PhD with as much ease and relaxed as you did. Even your colleagues who shared an office with you still don't know how you managed it. You never seemed stressed, except for the last few weeks, but somehow you always got the job done. After meetings and brainstorm sessions, driven by loads of coffee. You would head to the lab, and before you knew it, you'd come back with everything figured out. I must say, you almost seem the ideal candidate. And with almost, I really mean almost. And I think most people here, they know what I'm talking about. Because alongside your scientific achievements, you also you developed a very unique skill set. And that was the ability to terrorize the lab and your colleagues. I remember the day, it was April Fool's Day, and you set up a meeting together with Lydie that got Casper almost a heart attack. You sent pictures of boxes next to the -80 in the early morning with the message that you or someone else left them overnight and everything was thawed and ruined. It almost costed this man his life. And of course, there's the well-documented habit of leaving a selfie on the phone on anyone careless enough to leave it unattended for even a second. And I'm pretty sure some of your new colleagues had the pleasure to find them on their phones already. And let's not forget the infamous computer updates. I've heard some colleagues are still waiting for the computers finishing their fake updates. Let's say you were taught well. And of course, we cannot ignore the students that are also probably waiting outside of their offices because you decided to change the code from the key box again. And then, of course, there was countless dry ice bombs going off in waste bins on lab benches. Honestly, sometimes it felt we were in some kind of low-budget action movie. But let's not overlook your other obsession, and that's the one with tape. Of course, covering computer mice, so no one could figure out why the cursor wasn't moving. But speaking of tape, I also recall a more personal uses for it. And like you casually mentioned, that putting tape over some sensitive body parts before going for a run, because they were just too sensitive. Honestly, that was a level of oversharing none of us was really ready for.
I didn't share that.
You did. But in all seriousness, I've heard you in your new department, you finally met your nemesis. It seems that the universe had decided for a little payback. And despite all your pranks, you've been an amazing colleague. Your expertise, your technical skills, your writing skills, your willingness to help, your sense of humor, and of course, the endless coffees, chats, and the occasional beer. You've been more than just a colleague. You've been a friend to many. Some of those friends are standing beside you today as your power names, and one even made it to the love of your life. So maybe you really were the ideal candidate, after all. Joël, this chapter has come to an end, and you already started an exciting new step as a postdoc in the Department of Orthopedics. And I understand this is an important step you had to do for your career, and that you will do great and achieve great things. But when your contract ends, and I think that's next year, August, I hope you know where the good side of the hallway is. Congratulations once again, and thank you for everything you brought to our lab and the team.
Esteemed Dr. Beaumont, I'm here. Dear Joël, it's my pleasure to congratulate you with your doctorate, and I do that also on behalf of Maastricht University.
Thank you.
And I would like to share some impressions of the committee with you. You have well written, a good thesis. But it was an interesting but also a complex subject. But you managed well, and you have already published the majority of your studies in scientific literature. But it left also quite a number of questions left. And the committee has addressed quite a number of them. And we have seen in you as an enthusiastic, answering on an open way, a natural way, answer to the point, and still thinking while you were talking. So that was a nice way to see how you manage science. And we are very satisfied with your defense. I congratulate not only you, but also your two supervisors, Professor Casper Hoeschop and Dr. Tom Keulers. Congratulations with the result that we have seen today. I would also like to include my congratulations, your family, your partner, your parents, other members of the family. congratulations. And also my congratulations go to the audience here in the aula and also to the followers of the live stream. And I'm sure that quite a lot of colleagues and friends and other family members have enjoyed your defense today. I would like to thank all the members of both committee, the assessment committee, the thesis assessment committee, as well as this degree committee. Thank you very much for critically reading the manuscript, and I would like to thank also the external, in particular the external members of the committee who have contributed a lot to the committee, and Maastricht University appreciates your input. I would like to give the floor to Dr. Nolte het Hoen, who is still on the screen here, and I think she wants to congratulate you as well.
Yeah, Joël, so I already congratulated you on the thesis, and now I also want to congratulate you on a very great defense. I very much enjoyed our discussion. It was very insightful, and it was a good brainstorm session to generate more questions for the future of this research area. And I think it became really apparent that you're very knowledgeable on the research area that you worked on. So that is definitely worth a celebration. So I wish you a very pleasant day, celebrate well, and I wish you all the best for your future career. And well, hopefully we will see each other again in the future.
Thank you very much.
Enjoy.
Thanks.
Before I close this ceremony, I have one question and a remark. We will take a picture just after the closing of the ceremony with Dr. Nolte het Hoen at the screen, so we do that here. And after that we will go to the hall where we will take another picture, but there's no screen, so we have the complete committee now here. And if we go to the hall, I would like to invite the close family to go with us. And I would like to invite the other people here in the audience to go to the, so after the closing of the ceremony, you may go already to the rafter where there's the reception. You find it easily if you leave this aula and you make a turn at the elevator, you come to the rafter automatically. Hereby I close this academic ceremony.
All right, please.
We nemen eerst even de foto hier, Joël.
Ja, prima. Ja.
En als jij hier gaat staan met je twee vragen aan die we kunnen lezen.
Recording stopped.
Dat is dan allemaal, dat is, ja precies, als je moet je dan moet het boekje...